Nessa Carey, Hacking the Code of Life, How Gene Editing Will Rewrite Our Futures, Hot Science. Welcome to the world of gene editing, where our understanding of life's very blueprint is on the cusp of revolutionizing science, medicine, and agriculture. In Hacking the Code of Life, How Gene Editing Will Rewrite Our Futures, Nessa Carey takes you on a fascinating journey tracing the roots of genetic engineering back to the 19th century, through the groundbreaking CRISPR technology that now enables unprecedented precision, speed, and affordability in genome modification. From producing more robust crops to exploring novel therapies for genetic blood disorders, this book summary will provide you with an engaging account of how gene editing has the potential to reshape diverse aspects of our lives.
The Gene Editing Revolution. Scientists unveiled a new gene editing technique in 2012 that could revolutionize science and lead to major advances in medicine and agriculture. Dubbed the era of gene editing, this game-changing technology allows for the modification of organisms' genomes with unprecedented precision. Compared to earlier methods, the new technique is quick, inexpensive, and user-friendly. The evolution of genetic science. Long before genes were understood, humans crossbred plants and animals for optimal outcomes. Gregor Mendel's pea plant experiments led to Wilhelm Johannsen coining the term genes. Oswald Avery discovered that the genetic material was DNA in 1944. The double helix structure was then outlined by James Watson and Francis Crick in the 1950s, thanks to Rosalind Franklin's data. Learning how to manipulate genetic material has allowed for significant advancements in every aspect of human life. The evolution of genetic engineering. Scientists Stanley Cohen and Herbert Boyer made history with their discovery of genetic modification, enabling scientists to remove and insert genetic material from one organism to another. Though it was once expensive and challenging, modern breakthroughs in gene modification have made the process cheaper, easier, and more precise. Today, genetic engineering holds significant potential to treat diseases and improve food production, making it one of the most revolutionary fields in modern science. Gene Editing Revolution. The gene editing revolution began with the discovery of CRISPR, a technique that allows for precise editing of DNA. It all started with researcher Francisco Mojica, who discovered that bacteria had developed an immune system against harmful viruses by storing fragments of viral DNA between short, repeating sequences called spacers in their genome. Mojica and other scientists built on this discovery and created a way to use these spacers to snip apart the DNA double helix by using a molecular scissors to turn off or modify a specific gene. The precision of gene editing has gone so far as to allow editing a single letter of the billions in the human genetic code or regulate the amount of certain protein expression. This technique has drastically changed the field of genetics, allowing for more in-depth study of the role of specific genes and offering potential therapeutic benefits for those with dangerous mutated genes. Gene editing for sustainable agriculture. Gene editing offers promising solutions to address the challenges of global agricultural production while considering environmental sustainability. This technology has successfully reduced waste, removed potential harmful food constituents, reduced costs, boosted yields, and adapted to deteriorating environmental conditions. However, regulatory and consumer backlash may hinder the approval and market release of gene-edited crops. Global agriculture is facing a major challenge in meeting the needs of the growing global population while simultaneously ensuring environmental sustainability. This task is further complicated by developed countries' dietary habits, which include meat reliance and a culture of wastefulness. Gene editing, however, offers some promising solutions. Gene editing has already shown success in reducing waste by enhancing the appearance of fruits and vegetables without compromising taste or quality. It has also been used to remove potential harmful food constituents, such as celiac-triggering gluten proteins in wheat. Additionally, gene editing has enabled the production of cheaper substitutes for some costly ingredients, such as hops in beer brewing. Moreover, gene editing has increased yields while developing hardy crops that can grow in adverse environmental conditions. Rice yields, for instance, increased by up to 31% while maintaining its adaptability to harsh conditions. Despite the significant potential of gene editing in sustainable agriculture, the regulatory and consumer backlash against genetically modified, GM, foods remains a concern. Different countries have inconsistent approaches to regulating gene-edited crops. While the U.S. Department of Agriculture does not regulate gene editing if the modification could occur naturally, the UK imposes strict rules on gene-edited foods, similar to GM foods. Gene editing offers hope for sustainable agriculture, but regulatory and consumer acceptance remains a significant hurdle to overcome for the technology to achieve its full potential. Gene editing, a game changer for pharmaceutical industry. Gene editing is a powerful tool that can modify animals for human drug production and transplanting organs. Although the regulatory situation for meat from gene-edited animals remains unclear, it opens up possibilities for producing disease-resistant animals and those that can yield more meat. Gene editing technology offers a new era of sophisticated animal use leading to the creation of therapeutic drugs for human conditions. Pharmaceutical companies can leverage it to cultivate biological drugs that cannot be synthesized. Gene-edited pigs could serve as organ donors for humans in the future, which could revolutionize medical treatments. Gene editing advancements. Scientists are exploring the possibility of gene editing inside the human body to treat genetic disorders like sickle cell disease. They plan to remove stem cells from bone marrow, edit the cell's DNA, and reinsert them into the marrow to produce healthy red blood cells in patients. Another promising area for gene editing is the eyes, which are separate from the immune system and may be used to repair certain types of blindness. Gene editing can also improve cancer treatment by boosting patients' immune cells to fight cancer. Trials of this approach have resulted in successful cancer-free outcomes in 27 out of 30 patients. Despite the potential benefits, delivering gene editing reagents into the body and avoiding immune system reactions remain challenges for researchers and need to be addressed before further advances in gene editing can be made. Gene editing risks. Gene editing could pose potential risks. Scientists have raised alarm about editing genes that control cancer prevention mechanisms such as p53. Editing cells with weak p53 responses could increase cancerous cells. Additionally, techniques such as gene drives could cause unintended consequences. A gene drive could introduce a lethal mutation into mosquito genes and spread rapidly through the population. Although this could help in eliminating the pests, it would be challenging to stop the gene drives. Past experiences have shown that side effects from such changes are unpredictable. Gene editing and ethical considerations. Gene editing technology has the potential to prevent inherited disorders, but debates around informed consent and ethical implications arise. While gene editing remains theoretical, the risks and benefits of this technology, particularly in the human germline, require open discussions between scientists and society to ensure consistent frameworks across international boundaries. Gene editing technology has opened doors to the possibility of preventing inherited disorders such as Huntington's disease and Lesch-Nyhan syndrome, by editing the DNA of a zygote through in vitro fertilization. The corrected genome would then be passed down to future generations, raising ethical dilemmas around informed consent. Unlike standard medical procedures, gene editing procedures are performed on a bundle of cells without the ability to provide consent. The impact of these procedures on subsequent generations who have no choice in the matter is a matter of concern. While such interventions remain theoretical, discussions around the ethical and legal implications must increase. The consistent development of frameworks across international boundaries requires inclusion of all stakeholders. As gene editing continues to evolve, society and the scientific community must come together to explore the ethical and legal implications and provide guidelines for the responsible use of this technology. Gene editing companies attract billions. Investors have put in nearly $1 billion in gene editing companies such as Caribou Biosciences, CRISPR Therapeutics, and Adidas Medicine. The simplicity and low-cost applicability of gene editing technologies across species make it have a significant impact on basic science. However, the ownership of patents from the pioneers of gene editing, Jennifer Doudna of the University of California, Berkeley, Emmanuel Charpentier of the University of Vienna, and Feng Zhang of the Broad Institute in Cambridge, Massachusetts, will determine success. The patents could earn billions of dollars in royalties, and with a valuation of $1.2 billion and $2.6 billion respectively, Adidas Medicine and CRISPR Therapeutics could be the ones to keep an eye on.
In understanding the game-changing implications of gene editing, we've come to realize the transformative potential it holds for agriculture, medicine, and even the creation of disease-resistant animals. However, the path ahead is not without challenges. As the regulatory environment remains inconsistent, and consumer sentiment encompasses skepticism and optimism, scientists tread cautiously to avoid unintended consequences. While the future is uncertain, one thing is for sure: gene editing is rewriting the story of our collective future. As we navigate the complex ethical dilemmas and technological advancements, open discussions and cross-disciplinary collaboration will be key to ensuring the responsible and sustainable use of gene editing technology.